Welcome to the introduction to CellSim, a computing platform for growing virtual tissues to use in biological research. Before we begin the video demo, I'd just like to walk through a few introductory slides. Crowley-Davis Research set out to design and build a computing platform that would produce virtual tissues suitable for studying disease mechanisms, in silico toxicology, and so on. And this video demo is designed to introduce some of the important features of the platform and those of the virtual tissues. In this slide, we can see the overall organization of CellSim. Today we're going to focus on the ontogeny engine, which is shown here, which takes gene-level data inputs in the context of virtual cells and develops a robust metabolism and signaling between cells that gives rise to virtual tissues. Now, it's also true that you can do the reverse. That is to say, knowing the phenotype or the properties of a desired target tissue, we can use evolutionary search to find appropriate gene-level data inputs. But we're not going to focus on the evolutionary search today, but rather on development itself, which is the ontogeny engine. Now, we reasoned as we were developing this platform that to emulate ontogeny, the computing architecture had to parallel the organization of living systems. We call this a middle out, where the middle is in fact a virtual cell, and it controls genes, the molecules encoded by those genes, the interactions of molecules to produce metabolic processes in a networked way, either for metabolism or controlling gene regulatory networks. And at a higher level, the signaling between cells that gives rise to a self-organizing tissue. Now, what's important here, crucial really, is the list of features here that can arise when the computation platform is set up in this way. This next slide is simply indicating that the topic for study, the cell types we're going to study in this video, are B cells and T cells that arise from early lymphocyte progenitors. So this is a problem in lineage commitment. Here you can see a cluster of cells that differentiated into either B, blue, or T, green, from a few lymphocyte progenitors that grew, divided, and differentiated. Now, in these experiments, we also have a special STAT5 constitutively active transgene construct that can drive early T cell progenitors to become B cells by overexpression of the signaling pathway involving STAT5B. The next slide simply shows a portion of the networks for both wild type and the transgenic lymphocytes. Now, in the wild type, we do not have this transgenic pathway, which is shown here in the right hand, the middle panel, gene 2, which essentially goes directly to produce STAT5B without the need for signaling that is controlled in this portion of the network. Finally, in this last slide, we're just showing you some of the drop-down menus and tools that we're going to use in the video. The File menu, Tools, and Window, and the Resource drop-down menu. So now let's turn our attention to the video demonstration, and do some experiments with the virtual lymphocytes. This is part one of a video demonstration of CellSim. Today we're going to look at the behavior of wild-type lymphocytes. These are 5 wild-type cells in a sort of a grid pattern. And as the file name in the upper right indicates, these are cells that are going to grow and differentiate in a thymic environment. WT is wild-type. Now, in addition to examining the behavior of virtual lymphocytes, we'll also become familiar with some of the controls and tools and drop-down menus in CellSim. So let's get started with this. This main window here is called the phenotype viewer. Now, we have 5 genetically identical starting cells, virtual lymphocytes, and we're going to use pseudocolor here to track the concentration of intracellular components or resources. So here, the TN2 resource is a green color, very similar to what we did in the slides. And this green color will be used to follow or mark cells moving down the path toward T-cell fate. Now, if we click this resource drop-down menu here, we see that we also have a Pro-B option. And in this case, like we did in the slides, we're going to use a blue pseudocolor to indicate ELPs, early lymphocyte progenitors, that have chosen to progress toward a B-cell fate. The third color we're going to use is the ELP itself, and that is an orange color as shown here. So clearly, since with Pro-B, for example, these cells appear white, but with ELP, they appear orange, this indicates that these cells are in fact ELPs. So if we select a cell, let's choose this middle one here, you can see that some information appears here in the lower right. Now that cell ID of the middle cell in that grid pattern is ID number 147, and it has a substantial concentration of the ELP resource, nearly 191. So if we want to track then follow the progress of the simulation of development, we're going to press this start button in the upper left. And what we will see is that the steps, simulation proceeds in a stepwise fashion, and the steps will be increasing in both of these locations. The extreme lower left is the server, and this is what's being displayed against this black background. So let's let this simulation run for just a short bit until the cells divide. So we're going to click the run button, and you see that the cells are growing. We're at step 30 something, 50. The cells have grown. Oh, they've just divided. Okay, so I've paused the simulation at step 85. And if I want to, I can advance one step at a time. This is a finer control, so let's do that. Step 86, step 87. There we are. So we're at step 87 now. And you can see that all of the 5 original cells have divided. So these are the daughter cells that we're seeing now. And 3 of them, 3 of the daughter cells, appear white. Now, if we look at those individual white cells, let's click on one of them, cell ID 156. It has 0 concentration in the lower right here of the ELP resource. Cell 159, the same thing, and cell 150, also 0. So these cells, these white cells, the 3 white cells, are no longer ELPs. But what kind of cells are they? So we can go over to our resource concentration and choose a different resource. For example, Pro-B. So are they Pro-B? Well, apparently not. We can just look at a couple of cells quickly. Cell ID 183, 0. The other daughter in that pair, also 0, and so on. So these are not Pro-B cells. So let's do a quick inventory here to see if they might be TN2. So we'll choose that resource, the green color, and once again, these cells are white. They don't show any amount of TN2 resource in them. So I guess you could say these cells are in limbo. The white ones are. Some retain their ELP character, as we see here with the orange color, but these 3 white ones are in limbo. They haven't chosen a path yet for differentiation, but they have lost their ELP character. So since we're in a thymic environment, let's track the TN2 resource, which is what we might expect would be the fate of these cells heading toward a T cell fate. Let's track that resource and run the simulation forward, looking for the appearance of faint green color as these cells decide or determine to differentiate toward T cells. So here we go. Aha, okay. So we are at step 100, and you can see that those 3 cells, the ones that were white before, have indeed chosen to move toward a T cell fate. Excuse me. If we look at them individually, cell 156, we see it has a concentration of 2.35 of the TN2 marker in it. This one looks a little deeper green, and sure enough, it's 4.33 essentially, a little bit higher concentration of TN2. And this one at the bottom, cell ID 150, is in between the two, 3.57. So let's try to figure out what's going on here. First of all, what about the other cells, the cells that are not TN2? Well, all of those are orange. They are ELPs. So we can say this, that at this stage of the simulation, all of the original 5 daughter cells have divided, of the original starting cells have divided. So there are 10 cells total. Some of the daughter cells have chosen to move toward a T cell differentiation toward T cell state, and the others remain as ELPs. Now, this is what we we might expect in the thymic environment that we would get differentiation toward T cells, but we said at the beginning that the 5 original cells are genetically identical, and yet some have decided to move toward T cell fate, some have not, and the 3 that have decided, we just did the looked at the concentration of the TN2 resource and saw that they vary slightly. They differ from one another at this stage of the simulation. So why, if they are genetically identical? Well, the simple answer is that these cells are responding to their local signaling environment. And so we might think that this pair, which had the highest concentration of TN2 in the lower cell, is receiving a stronger signal than the other 2, and that these 2 pairs of cells in the middle and the upper don't have any T cells or TN2 character in them, so they might be experiencing a little bit weaker signaling. So it's true that the signaling strength varies slightly across the field of view. Now, so what we might want to know is, are these cells, the ones that have not committed, are they going to commit? So we need to move deeper into the simulation. And we have an option up here that instead of watching the steps advance one by pressing the run button, we could do that. But if we use this other control called advance to step, we can choose to advance the simulation to a predetermined step. So I'm going to say step 200 here. And when I press this button, you will see in the lower left that the step numbers on the server side will advance very fast, and there won't be any change in the display here until we get to step 200, and then it will show you what the cells, what the field of cells looks like at that point. So let's go ahead and press advance to step. Very rapid advancing, and you see that there are 13 cells now. Some of these cells have divided. And in fact, one of them, this upper one of the cells is no longer an ELP. And in the grouping of cells on the right, there are 2 cells that are no longer ELPs. And finally, the center pair of cells are still both ELPs, that is, neither daughter cell has committed to leave the ELP state and either enter a path towards a T cell or a B cell. So let's look at these 4, 1, 2, 3, 4, 5, these 5 white looking cells that are not ELPs. Are they pro-B? Well, no, they're not. They don't express any pro-B character. Are they TN2? Yes, they are, and that's what we would expect. So this cell was one of the first that we looked at, and its concentration of TN2 is way up there now, 76. It's fully committed there. This one is only 1.4, so it's just recently started to commit to the T cell fate. But we saw that none of these is pro-B. So let's try this again. Let's move further into the simulation using the advance to step button and see if this pair of cells in the middle eventually commits. So let's advance to step 500. The server rapidly advances. Boom, there's step 500. We have 21 cells, and you can see, yes, in fact, there are TN2 cells, and the middle cell in the original grid of 5 has divided. One of the daughter cells has divided, and that daughter cell has now committed to a T cell fate. The rest of the cells in this field of view are ELPs, as shown here by this the orange pseudo color, and we don't see any evidence of pro-B cells. So each of the original 5 cells in the original 5 ELPs, group of 5, has produced one or several TN2 cells, one in this case, one here, 4 here, 1 here, and 4 there. And that's as we might expect for a thymic environment. So let's summarize these results in a slide that is simply a composite of screenshots. So I will call up the slide here, and we can see that at step 100, we have some early cells, early daughter cells, light green in color, going down the path to TN2, eventually to T cells. We use step 200, but here showing you step 300, more TN2 cells and deeper green color. And in the final stage that we looked at, step 500, we see that there are no pro-B cells. There are plenty of ELPs still there to generate T cells, TN2 cells, and we see quite a few here. Now in the next sequence, we're going to dig a bit deeper and do some experiments with transgenic virtual lymphocytes. Welcome to the second segment of the video demonstration for CellSim. In the first segment, we looked at the behavior of wild-type virtual lymphocytes in a thymic environment. And here we have a similar grid pattern of 5, except these are transgenic lymphocytes, as this file name indicates. So once again, a thymic environment now, but transgenic lymphocytes that have a pathway for constitutively active STAT5B component that can drive ELPs, or more properly ETPs, early T cell progenitors, toward a B cell fate, even though they're in the thymic environment. Now you may recall that in the previous simulation with wild type, we saw no B cells even out at step 500. So we're going to use the same procedure here. We're tracking TN2 in green, ELP in orange, and pro-B in blue. I'm going to advance to step 80. And we can see that there are no cells at this stage committed to the T cell fate, but there are in fact 10 cells. They all have divided once. And if we look at the ELP, it's very similar to what we saw with the wild type cells. They are all still ELP cells, and we don't see any evidence of pro-B yet. So let's do what we did before, advance to step 200, and examine the field of cells again. Now we have 13 cells, and indeed, unlike what we saw in wild type, we now have 3 cells that have begun to commit or are committing to the B cell fate in this thymic environment, indicating that indeed these are transgenic virtual lymphocytes. Now, what about ELPs? Well, there are a number of those, and finally, T cells, or TN2 stage. Yes, in fact, there are 1, 2, 3 cells that appear to be heading down the pathway to differentiate into T cells. Now let's go back. Let me pause for a minute, and I'll be right back. Now, some of you may have noticed that this cell over here in the right-hand grouping, this upper cell shows a little bit of B cell character. It's an early pro-B, and it also shows a little bit of T cell character, light green. So let's advance to step 500 and see which fate that cell chooses. Since it has a little, it seems to have options. So we're advancing rapidly here to step 500. And we need to look at cell number 425. I should have pointed that cell ID out before. And it's not a TN2. It's not an ELP. It has committed solidly to the B cell fate. So this little demo here should serve as proof of concept for the efficacy of this virtual STAT5B constitutively active pathway that will drive cells toward the B fate even in a thymic environment. Now I'd like to do some more experiments to dig a little bit deeper, and so I'm going to pause the simulation and come back with a little bit different experimental setup. All right, I'm back from making a few modifications here, and as you can see in the upper right, I still have the thymic environment, transgenic virtual lymphocytes, but the file has been modified, and I'd like to show you what those modifications consist of. For that, we're going to use some other tools and drop-down menus that we haven't seen before. So I'm going to use a window here that is called the Gene Assembly Editor. And what this Gene Assembly Editor allows you to do is to change the way the network is structured in one or more cells. So if we're looking at cell 495 here in this menu in the Gene Assembly Editor, well, which cell is that? It's the cell in the on the left-hand side of the grid of cells. So back to the Gene Assembly Editor, 495, we can see that there's a positive 0.8 promotion value that this is the transgene that produces the STAT5P. But if we look at another cell, any of the other 4, we can see that in those cells, let's look at number 492, this transgene has been turned off by me by manipulating this value, putting it at 0, and we see that the other 4 cells, 489, the same thing, it's off, 486, it's off 498, the transgene is off, and in 495 again, it is on with a positive 0.8 promotion value. So that would indicate that these transgenic lymphocytes, only the descendants of cell number 495 are probably, we would expect, going to behave like they are transgenic, and these others might behave like they are wild type, because I've turned off or done a complete 100% knockdown of the transgenic pathway. Now I'm going to, in cell number 495, I'm going to add something here that when that is turned on, I'm just going to call it marker 495, and it's going to produce this unique marker that will mark that cell and all of its progeny. So let's upload that and run the simulation forward in much the same way that we did before. But before we do that, let's just make sure we understand here by looking at the metabolic network for cell 495. Make sure that we understand what we're talking about here by looking at this upper portion of the diagram. And we can see here is marker 495 that we just added. Gene 2 is the bypass that goes directly to make STAT5P without the need for IL-7 receptor-mediated signaling. And let's choose another cell just to show you what all the other cells look like. Again, this one has a weak positive promotion value, so it is going to make this marker and it is going to be transgenic. And if we look at any of the other cells, 489, for example, it has to redraw this. We see there is no marker in this cell, and this has been turned off. So indeed, we can use these tools to make sort of a custom side-by-side comparison in our field of view to do our experiment with virtual lymphocytes. So I'm going to repeat the same sequence of going to step 80, examining the cells there. And you can see that there are 10 cells. We knew that. Are they all ELPs like we've seen in previous? Yes, they are. But now in our resource catalog, we have a marker that is the marker for 495, and that the default color chosen is orange, and we don't want it to be orange, so we won't get confused with ELP. Instead, we'll make it hot pink. Here we go. So that all of the progeny of this cell are going to be marked in that way. So let's advance to step 200 and see what we're getting. Now we can see that there are 4 progeny of I'm just rotating the cluster of cells so that you can see better that there are 3 cells that are descendants of the original cell number 495. And one of those is a B cell. So we're already seeing some evidence that the transgene is working in these and it's not working in the other 4 because I did a complete knockdown or turning off of that transgenic construct in those 4. So, excuse me, have to get TN2 marker. We see that there are some T cells again here. And if we advance to step 500, let's see if the picture remains consistent here. Step 470, there we are, step 500. So what we have, if we look at this left-hand cluster of cells, all of those are descendants of the cell with the active transgene. The other cells are not. They are descendants of the other 4 starting cells. Those other 4 starting cells in their progeny all produce T cells, but as did the cell number 495, but only cell 495 produced B cells. So this shows you how easy it is to be able to look at growth and differentiation in virtual tissues, to track the progeny of individual cells, to manipulate genes, both the products they encode and the promotion values thereof, to add new gene assemblies. We didn't do that, but it's possible to do it, and to examine the gene regulatory networks, signaling networks, and metabolic networks of individual cells while doing experiments. So that concludes our demonstration of the transgenic virtual lymphocytes.